All right, so this presentation is just an update, a bit of a recap of what the project is about, the things we found in previous years, and then a summary of the results that we found this year. We were lucky to be able to continue research quite intensely, despite COVID measures. That was a good thing. So first, I would like to, acknowledge my co-applicants, Sarah and Siobhan. They're essential parts of this project as well, with all their experience and advice. We're all in this project together. So the whole premise was what kind of early intervention points can we determine and research to see if we can make those into IPM recommendations. So our goal is to keep pests such as whiteflies and thrips at low levels while still growing nice plants, of course. So the first objective of this project, which we already finished, was dipping cuttings. To reduce infestations of pests on imported propagative material. We've shown that cutting dips basically knocks down pest populations right at the start. It's indicated here, if you can see my mouse moving over the slide here. So right at the beginning, we already lower pest populations that may come in on plant material, so that it wins time for the biocontrol agents to start working. And this is compared to the red line. If you would not dip, pest numbers would theoretically increase like this. And if you dip, pest numbers would be set down, but eventually, if you don't do anything else, they would still go up. And this objective has been completed. Now, the next objective investigated the effects of plant nutrition on pest populations. And especially nitrogen has been identified as an important element for insect nutrition. This diagram kind of tries to describe that. Nitrogen is basically taken up in the roots here as nitrate and ammonium, and it's transported throughout the plant, these arrows here, in the xylem and the phloem as amino acids. And nitrogen then also can be stored as inorganic, so again the nitrate and the ammonium, or as organic nitrogen like amino acids and proteins. So why am I talking about plant nutrition while I'm a pest person? Basically, the nutritional value of plants for pests is in part determined by the organic nitrogen, the amino acids and the protein. That's their food. That's where they get their energy and their nutrients. Whiteflies feed on the phloem, and thrips feed on the cell contents, and there they encounter these amino acids and proteins. So by manipulating the amount of fertilizer we give to the crop, we can potentially change the plant's nutritional value for pest species. And as in Barry's talk, you've already heard, floriculture crops are often over-fertilized. So there's actually room to play with this and make this maybe an IPM strategy. So the research in objective 2 of this project is to determine if reducing fertilizer will slow down pest population increase and actually lead to better biocontrol performance. Of course, you can reduce nutrients too much as well, which will negatively impact plant quality, which is why we put in an objective also about biostimulants. Biostimulants are beneficial bacteria or fungi or extracts of those, and these may mitigate these negative effects while enhancing host plant resistance. We don't know a lot about biostimulants, but we know that they help plants deal with reduced nutrients and that they can also induce plant defenses against pests. And that's summarized in this graph here. So the microorganisms, the beneficial microorganisms here at the root zone can have effects throughout the plant that can benefit the plant in multiple ways. So the whole idea is to optimize plant nutrition to reduce pest population growth, and then that the residual populations will be managed by biocontrol. And that's the, if we go back to that graphic that I showed at the beginning, the blue line would be this theoretical situation where we dip cuttings and we reduce fertilizer to a level that it benefits less the pests, but that we still produce a nice plant. And then, of course, we could still add biocontrol because, again, populations may still increase a little bit, but less fast. So your biocontrol success would be higher. So this is the actual objective that we put in a proposal. We want to optimize nutrient inputs and biostimulant amendments to reduce the risk of pest outbreaks while manipulating plant health and quality for two major floriculture crops. We chose Gerbera, potted Gerbera, and potted chrysanthemum. And the two pests that we chose for each of these crops, for Gerbera, we chose white greenhouse whitefly, and for chrysanthemum, we chose western flower thrips. And then for the whiteflies on Gerberas, we also wanted to see if we could quantify the effects of reduced nutrients or biostimulants on biocontrol efficiency by entomopathogens. So this slide summarizes the experiments we've done on whiteflies on potted Gerbera. So we varied the different colored bars. Here's the legend, represent different rates of fertilizer. We cut the whole fertilizer in order to obtain rates of 25 ppm of nitrogen, 50, 100, 150, and 200 ppm of nitrogen. And we confirmed that this actually had an effect on the organic nitrogen levels in the plants by sap analysis. And sap analysis is something that really gives you a snapshot what's happening in the plants right now. And because the whiteflies and thrips as well feed on the plant sap, this is actually their nutritional shake that they encounter. So we saw that plant quality was affected at about that middle rate, the 100 ppm of nitrogen, and that, of course, whitefly populations increased over time. And here you see the ANOVA table of the statistical analysis, and that's a significant, there were significant differences over time, obviously. But what we were more interested in is if there were significant effects of treatment, those different fertilizer rates, did they actually result in different whitefly populations? Unfortunately, we, over this time frame, this is basically from transplanting the Gerbera plugs to flowering, period of about 10 weeks, we did not see any significantly different, statistically significantly different effects of fertilizer treatments on whiteflies. I mean, you do see kind of a few differences, but if you look at these error bars, you can see that there were statistically no differences. So it may be that if this would have gone on for a much longer time, we would have seen some differences. But basically, the life cycle of whiteflies is about 1 month from egg to adult. It's too long compared to the crop cycle to see any effects. Fertilizer levels in the plant, or sorry, nutrient levels in the plant don't kill whiteflies, right? It just slows them down. So that's why we probably did not see any effects in whiteflies on potted gerbs. Now, we still had a chance. We put our hopes on this theory that reduced nitrogen could increase whitefly susceptibility to entomopathogenic fungi. There's literature that suggests that insects are better able to resist infection by entomopathogens when they feed on a diet that is high in nitrogen. So if we lower that, we would hope that maybe they would be more susceptible. What we did was we infested plants with clip cages with whiteflies. So we looked at the second and third instar nymphs. We grew the plants under low fertilizer, 50 ppm, versus high fertilizer, 200 ppm of nitrogen. And the biocontrol was botanic art wettable powder at different rates. So we took the full label rate, and then we started cutting label rates up to until 10% of the recommended rate, just to see what would happen if we challenged them with a little bit less than the recommended rate. Even at the lowest rates, so here, of course, the control whitefly mortality was really low, but even at 10% of the recommended rate, actually whitefly mortality was pretty high, and there was no difference between low and high fertilizer rates, even at the lowest botanic art rate. So unfortunately, that didn't work out as we would have hoped, or that was our hypothesis. Now for thrips, the story is quite similar. Again, we did several blocks of the same trial where we varied fertilizer levels. These again represent different levels of ppm of nitrogen. This for chrysanthemum, we took 25, 50, 100, 200, and 300 ppm of nitrogen. And again, this is over time how thrips numbers responded. We did again sap analysis. So yes, there were different organic nitrogen levels in this plant sap. And again, plant quality was affected at around the 100 ppm level. As you can see from the ANOVA table, again, over time, thrips populations increased significantly. Not a surprise, because that's what thrips do. But treatment, and this was the frustrating part, here we have a very marginal effect. It's officially you want to see 0.05. That's the standard of what you define as being significantly different. Here we see 0.088. So we could consider this a trend. So there may be some fertilizer effect, but we would have to do either more replicates or change other things to really prove that this is a thing. There was a very high variability among the different blocks that we did in summer and winter and spring due to environmental factors, we think, and that's why these error bars are pretty big. And maybe that's what's made it so difficult to prove that we could have maybe significant differences. Now, a very interesting observation was we did have an accidental infestation of two-spotted spider mites, despite the fact that we released Persimmilus, and they seem to also have a very strong response to fertilizer in one of the trials. So I really still think, despite these results, that fertilizer nutrient levels in the plant actually affect pest populations. We just have to prove it. Now, we'll come back to that later as well in the discussion, but first I want to talk about the biostimulants. So our theory was that biostimulants could maybe help make up for any effects of low nutrients on plants, right? They help plants cope with low nutrient levels. So we tried two different biostimulants and confirmed that they actually were present on the plant roots. And we did this under high and low fertilizer levels. So again, 50 ppm and 200 ppm. There was some effect of one of the biostimulants on root length under the conditions that we grew them under, but basically the selected biostimulants did not seem to compensate for low fertilizer treatments under our conditions, under our fertilizer rates. The effects on plant quality were only due to high and low fertilizer. So obviously on the high fertilizer, you see more dense foliage, higher foliage mass, and more flowers. And on the low fertilizer levels, we saw increased root length and root mass. Now, the thing we were after as well was to see if biostimulants could have any effect on Thrips populations, maybe through induced plant defenses. So again, on Thrips populations, we did not really see any effect of the biostimulants at either low or high fertilizer. So the biostimulants did not seem to do anything for the Thrips populations. And now the Thrips populations, again, were quite variable, so it was very hard to draw conclusions. But we did, again, see some effect of low versus high fertilizer on Thrips populations. So that, again, reinforced our gut feeling that there is something going on. And these two graphs here, the blue is low fertilizer and the orange is high fertilizer. At the end of this trial here, you can see that there's more Thrips in high fertilizer level treatment. And here, again, this was actually statistically significant again. So we're not completely ready to give up on the fertilizer levels for Thrips. Although the biostimulants, we may have to try different biostimulants or reduce nutrients in a different way. So in conclusion, basically the whiteflies on potted gerbera, we did not see an effect on whitefly populations within the time frame of a crop cycle of about 10 weeks. And also we did not see any whitefly susceptibility to biopesticides differences. For the Thrips on potted chrysanthemum, and again, I'm couching this in really careful terms, there's a possible effect, a trend only, of plant nutrition on Thrips populations. And the differences between those two bars is actually up to 50%. So it could be, it could help in an IPM program. We are still testing the combined effect of reduced plant nutrition and biocontrol for Thrips as well. And this is supported by the American Floral Endowment as an add-on to this project. But these trials are still ongoing. So at this point, we hope to have recommendations that we could validate in commercial greenhouse tests. Instead, we're going to do more lab studies, really to dig deeper into what does nutrient levels in the plant do to Thrips populations, to Thrips life history parameters like development time and that kind of thing, oviposition rates in a more controlled environment. And we also want to try to see if chrysanthemum cultivar has an effect. So we're just going to do more trials to dig a little bit deeper into this question. Because other studies also have some proven effect. But we think maybe there may be kind of an iceberg effect going on, that only the studies that show an effect get published, while the ones that don't show an effect don't get published. So maybe something that's going on. Other things that we don't know is what is going on in the plant in response to fertilizer rates with regards to metabolites, plant defenses, that kind of things. And also in this study, we cut the complete fertilizer rate, but we don't know what happens if we only change the nitrogen, for example. But that would be a multi-year experiment as well. So if these lab trials work out quite well, then hopefully we'll still have some time to validate results in commercial greenhouses. Hopefully we can do this in Quebec and Ontario and incorporate best practices from the dipping and reduced fertilizer rates. Over the past year, we managed still to do some outreach. These are all results that were integrated in other presentations. So we gave 3 presentations here. And we did a webinar for Crop Talk Media that's actually available online as well. So with that, I'd really like to thank all the people here who worked very hard to make all these trials happen. It's a lot of work. So Ashley, Zelda, Taro, and Parker, and the Vineland Greenhouse staff, I would like to thank them very much, as well as the funding partners. So thank you for listening. And if you have any questions, I'll be happy to try and answer.